All right, I think people are coming, so maybe we can start. Then or just join us, then they can later come, right? So, hello everyone. Welcome to one more webinar. My name is Renata Daft. I'll be your host today, together with my colleague Thomas. So we are very happy to have all of you with us today. So I'm very happy to have two excellent speakers with us, Professor Alessandra Sussulini and Henrique Camacho. We are going to start with Professor Alessandra, so I'm going to introduce her. So Professor Alessandra is Associate Professor at the University of Campinas, Universidade Estadual de Campinas, Unicamp in Brazil. So she is head of the Laboratory of Bioanalytics and Integrated Omics, called LaBIOmics. She also did her Ph.D. at the University of Campinas and a sandwich period at the Max Planck Institute in Germany. She completed her postdoctoral training at the Butantan Institute in Brazil and a research center Jülich in Germany. At the moment, her focus is on mass spectrometry, multi-omics integration with application in neuropsychiatric diseases, molecular mechanism, and natural psychoactive compounds, and the characterization of agroecological foods. She also has authored over 70 peer-reviewed articles, 9 book chapters, as the Spring Nature book on Metabolomics in 2017. And more recently, she received an academic recognition award in human rights and the Vladimir Herzog Institute in 2023. And last but not least, this month she was highlighted in the cover of the Metabolomics journal as one of the most influential women in metabolomics. So Professor Alessandra, thank you for accepting the invitation. It's a real pleasure to have you here. And the floor is yours. You can start your presentation.
Thank you, Renata. Thank you for the kind introduction. I will share my screen now with you. Okay, so thank you again for the invitation, all the Early Career Members Network for the invitation to be here. It's really a pleasure. And I'll talk about some works we have been developing in my lab. I call this presentation as a metabolomic perspective on neuropsychiatric disorders and natural psychoactives. So first, I would like to show you where I come from. So I work at the University of Campinas. Here on the left figure, we can see the entrance of the Institute of Chemistry, where my lab is located. And on the right photo here, upright, we can see an aerial night photo of the whole campus, which has the same shape of the university logo. Our university is relatively young. We are turning 60 this year in 2026. And we are located in the city of Campinas, the city where I was born. And the city is considered one of the biggest ones in Brazil. It is on the top 15. And it is located 100 kilometers far from São Paulo, the biggest city in Latin America in terms of population. And the population here in Campinas is above 1 million people. So we are a quite big city. The city is very diverse. We have some historical buildings. We have also modern ones, and we are very surrounded by nature too. So this is my research group. I would like to start showing my research group because sometimes it stays at the end of the presentation and we don't have a proper time to talk about them because without our research group, we are nothing. So I'd like to start thanking the current members and the former members of this group. This is our most recent photo. We were established in 2014 at the University of Campinas. And here is our social media address and our webpage, so you can follow our updates in these channels. So to start, I would like to show you a bit of our research timeline. So I started to get interested in studying psychiatric disorders back then in 2007, when I was a PhD student. And I got very interested in working with bipolar disorder. So I started the first works in this disease here in Brazil back at that time. So in 2015, already in my research group, we also got interested in studying another diseases, and one of them is depression. And more specifically, we wanted to understand potential novel treatments about this disease. And we started to work with, at that time, with the psychoactive substances present, for example, in ayahuasca. And more recently, in 2022, we also got interested in studying neurodegenerative diseases and the potential treatment with cannabis, another natural product. So first, I'll talk a bit about bipolar disorder research. I'll not be too long here because Henrique, the next presenter, was my master's and PhD student, and it was the topic of his work. So I'll talk very, very briefly about the first and the last study we have published on this topic. So bipolar disease is characterized by mood oscillations, where the patients have experienced episodes of mania, also called as highs, and episodes of depression. These episodes can be shifted very quickly, and they are intercalated by periods of the so-called euthymia, the normal mood. And the stabilization of the mood is usually performed with the treatment with mood stabilizers. The most famous one is lithium, which has been used as a gold standard for bipolar disorder since the 1950s. And these mood stabilizers are usually associated with other drugs, for example, antipsychotics, antidepressants, and so on. The causes for this disease are not fully elucidated. There are some genetics, neurological, and environmental factors associated, but we don't have a complete answer so far. And the diagnosis for bipolar disorder is based only in a clinical setting, in a clinical interview between the psychiatrist and the patients. So it's not easy for the psychiatrist to give a precise diagnostic for the patients because they don't have any other type of tests, blood tests, for example, to provide this proper diagnostic. So metabolomics enters here as an alternative to understand better the molecular mechanisms behind the disease and propose biological signatures that can help to elucidate its pathophysiology and also give us hints that can lead to further diagnostic tests. So the first study was published in 2009 during my PhD. At that time, I did not work with mass spectrometry, which is our specialty nowadays. This first work was performed with NMR spectroscopy because it was the technique we had at our hands at that time. And in this work, we used serum samples from 3 different groups. We had a control group with healthy individuals. We had a bipolar group of patients treated with lithium. As I mentioned before, it is a gold standard for mood stabilization in bipolar disorder. And another group of patients with bipolar disorder, but treated with other drugs except lithium. And then we just diluted the serum samples with D2O. We acquired the NMR spectra for each one of the samples. And then we performed data pretreatment with orthogonal signal correction, and afterwards multivariate statistical analysis by interval PCA and PLSTA. So here is the summary of the main results of this work. So this is the PLSTA scores plot. On the left side, we can see the control group samples all together. And on the right side of the plot, the bipolar patient samples. And we could also observe the grouping of the bipolars treated with lithium or with other drugs. And by the loadings plot results, we observed that the main differential metabolites to perform this differentiation between the groups were the lipids, more specifically glycoprotein lipids and monopolysaturated fatty acids. We also observed some lipid metabolism-related molecules, such as choline and myo-inositol, and also some amino acids, such as glutamate, lysine, and arginine. So this first work was important to show us that the lipids were very important for this differentiation between the healthy controls and the bipolar patients. And further works were developed using the lipidomics strategy. I will show now the most recent published result about lipidomics, but we had some differences here because in the case of this study, we had plasma samples instead of serum, and we have again 3 groups. But in this case, we had a control group, a group of bipolar patients, and a group of schizophrenia patients. Schizophrenia is a very interesting psychiatric disease because it has symptoms very similar to the mania state of bipolar disorder. So they share common symptoms, both diseases. And in the case of this study, all the patients were drug naive. The samples were collected when the patients were admitted at the hospital. At that time, they were their first psychotic episode. So they did not have a close diagnostic by so far. So after a following up of these patients at the hospital, they received the differential diagnosis, but at the time of the sample collection, they were just at their first psychotic episode. So these plasma samples underwent lipid extraction using a 3-phase extraction with MTB, methanol, and water with ammonium acetate. The organic phase was analyzed by LCH-RMS using an Örebro system. And after that, we performed data analysis and biological interpretation. So here on the left figure, we can see the PCA scores plot for the samples. In red, the bipolar samples, in green, the control ones, and in light blue, the schizophrenia. In dark blue, we can see the quality control samples. They were all clustered together here at the center of the PCA scores plot, showing us a very good instrumental stability. And on the right figure, we can see the heat map of the differential lipid species determined by ANOVA. So here we can see in red the bipolar samples, in green the controls, and in blue the schizophrenia samples. Just by looking at the heat map, we can see interesting differences between the groups, especially in the schizophrenia group where most of the lipids appeared with a lower concentration level. Taking together the information about these lipid species, we classified them according to their major subclass. So here this graphics shows us that. So when we compare the groups 2 by 2, we could see some tendencies. So when comparing the bipolar samples with the controls and the schizophrenia samples with the controls, the distribution of the main differential lipid classes was quite the same. So the main differential lipid class here, when the comparisons of the controls versus the diseases, were the glycerophospholipids. But when we compare both diseases, bipolar disorder versus schizophrenia, this profile changed a bit. Now, the main differential lipid classes is related to the sphingolipids and also the glycerolipids that appear with a lower importance when comparing bipolar or schizophrenia versus the controls, almost double its importance here in the comparison between the two diseases. So it was very interesting, and it raises some hypotheses for further studies on validating and proposing differential lipid profiles of the two diseases. We also performed the ROC curves for these differential lipid species, again comparing the groups 2 by 2. So when we compare the bipolar versus the controls, we had at the total of 90 differential lipid species. And for the ROC curve, we got an area under the curve of about 0.7. When comparing the schizophrenia patients versus the controls, we got a total of 44 lipid species, differential ones, and an area under the curve of about 0.9. And comparing bipolars versus schizophrenia, we got a total of 23 differential lipid species and an area under the curve of 0.8, showing that these models have a potential for discrimination about related to these comparisons we performed. So it was very interesting. And nowadays, I have a PhD student working on that because we are also performing targeted metabolomics and lipidomics for these metabolites and lipids. We got information about this potential for differentiation, and we are looking forward to a validation of these results in order to improve bipolar disorder diagnosis, and also proposing a differentiation between bipolar and schizophrenia patients, which will help a lot the psychiatrists to perform a more precise diagnostic and the patients to have a better quality of life with proper treatments. So moving to another psychiatric disease we studied in our group, I'll talk a bit about depression. It also has some symptoms very close to bipolar disorder in the low state. And depression is characterized more by sadness, loss of interest in many activities, suicide thoughts, and sleep disorders. The main issue about depression is that only half of the patients respond to the ongoing, the conventional treatments. 32% respond only partially, and 44% do not respond at all. So these patients are known as having treatment-resistant depression. The treatment for depression is usually performed with antidepressants associated or not with psychotherapy. The causes are related to genetic, environmental, and personality factors. And again, as for bipolar disorder and schizophrenia, we don't have a blood test, for example. We only have a test for thyroid problems to discard them because the symptoms are very close to the depression, but it's not a specific test for depression. And again, it's important to understand its pathophysiology and look for biological signatures of depression. But as I mentioned before, the main issue about depression is related to its treatment. So the treatments we have available nowadays are not fully effective. So we started to look for another types of treatments. And one that appeared very interesting was using ayahuasca. So the basis for this interest was regarding this work published in 2015 by this group of researchers here in Brazil. They observed a very significant decrease of depression scores using two different depression scales, the HAM-D and the MADRS, and they observed that these patients with a single dose of ayahuasca produced a decrease in these scores. So these are the results here for 1, 7, 14, 21 days after the ayahuasca intake. They present this decrease in the scores, and we got very interested to understand what's going on. Is ayahuasca really effective for treating depression? So we wanted to use metabolomics to understand that. But first of all, the first question, what is ayahuasca? Ayahuasca is a traditional Amazonian beverage used by the indigenous people here in South America and Central America too, by millennia. And it is used in healing and spiritual rituals, and it's known for having psychoactive effects. So ayahuasca is prepared by the decoction in water of these two plants, Banisteriopsis caapi and Psychoactives viridis. And what is very interesting about that? The leaves of Psychoactives viridis have this psychoactive compound, and dimethyltryptamine, DMT. And you might think, why not using only the leaves to prepare ayahuasca? That's why what's happened here? Ayahuasca, with the DMT present in ayahuasca, when ingested, it is degraded because we have the monoamino oxidase and it degrades DMT. It doesn't have any psychoactive effect when ingested. But when this other plant is mixture, this plant have these beta-carboline alkaloids, which are monoamino oxidase inhibitors. So then these alkaloids inhibit this enzyme, and then DMT has its psychoactive effects. This is very interesting, how the indigenous people realized that they needed to mix the two plants to have any effect. We will never know. This is a mystery for us. So this is a very interesting natural product at all. So the first study we performed, we wanted to understand better what is ayahuasca before thinking about it as a treatment. And the first strategy was a targeted metabolomics one. We developed and validated a method for the quantification of the four main alkaloids in ayahuasca, DMT, and the other three beta-carboline alkaloids present in the Banisteriopsis caapi plant, harmine, harmaline, and tetrahydroharmine. In this case, we used a triple quadrupole mass spectrometer and a selected reaction monitoring strategy. So here I show some results we got because we developed, validated the method, and applied it to two sets of samples. The first set of samples was acquired here in the São Paulo state. They were all prepared by the same religious group, and we observed some concentration ranges for all the four alkaloids. And we also applied this methodology to a set of samples acquired in Europe. So these samples were sent to our lab. They were acquired in different countries, and they were prepared in different contexts, because not only religious ones, but also recreative. So here we can see a very dispersed concentration range. It is reflected here by the mean values. For example, for DMT, almost 4 times higher than for the ones, the samples acquired here in Brazil. But we can see that for some samples, we have almost no DMT. And for example, for harmaline, some samples had no concentration of these alkaloids. So it raises our concern. Are these samples all ayahuasca? Then we applied an untargeted metabolomics strategy to evaluate the chemical profile of the samples. So for most of the samples, we got a profile very similar to this first chromatogram. So we can see here the 4 main alkaloids and other compounds present in ayahuasca naturally. But for some samples, the profile was very odd. I have shown us, I'm showing here 2 chromatograms that raised our concern. The second one, we can see only harmine, the only alkaloid we have found that was expected. And we observed a very intense peak here that we, using HRMS, we determined that it was mescaline. And mescaline was not supposed to be here at all because it is present in peyote. It is a cactus. It is also psychoactive, but this is not ayahuasca. And for the other sample, I'm showing here the chromatogram. In this case, we can see the 4 main alkaloids of ayahuasca, but we also found different molecules. We found uremamine, which is present in another type of plant, the Jurema preta. It also has DMT, but is not Psychotria viridis, the plant traditionally used to prepare ayahuasca. We also have found psilocin here, which is present in mushrooms. It is a psychedelic, but it's not supposed to be in ayahuasca. And we also found moclobemide, which is an antidepressant, a synthetic drug. So this raised our concern, and this showed us the importance of quality control in ayahuasca for thinking about further therapeutic purposes. In a more recent work, we then we characterized ayahuasca, and now we wanted to understand how it acts on a cellular level. So for this study, we had a primary cell culture of astrocytes, which are glial cells that support neurons and regulate brain functions. And we applied to the cells ayahuasca in different concentration levels. Since ayahuasca, we have seen it is a complex mixture. So we needed to choose one molecule to standardize the concentrations. We have chosen then DMT, the psychoactive one. So we applied different ayahuasca concentrations in terms of DMT to the cells. We collected the intra and extracellular content, and we analyzed it by LCH-RMS using a Q-TOF instrument and hydrophilic interaction chromatography for separation. So the first result I'm going to show you is about the cell morphology obtained by microscopy. We could observe that the cells treated with different ayahuasca concentrations showed at least slightly changes in the cytoplasmic volume. We tested here 10, 20, 30, and 40 micromole per liter in terms of DMT. For the concentrations above 20, we observed an increasement in cell death, and above 30, a presence of apoptotic bodies in the cytoplasm. So this result showed us the importance of testing different concentration levels. We also performed the viability assays by MTT. We applied this reagent. When the cells are viable, they reduce it to formazan, and then we measure it by absorbance. So we performed some positive and negative controls, a control with ethanol in different concentrations as positive controls of cell death and genotoxicity, and afterwards we tested the different ayahuasca concentrations. And again, these results confirm the ones we already observed by microscopy, that for concentrations above 20, the cell viability is lost in a huge intensity. Therefore, for further metabolomics studies, we decided to work with lower ayahuasca concentrations. Then we tested 1, 10, and 20 micromole per liters in terms of DMT. So summarizing the main results for the metabolomics analysis, the main intracellular metabolites altered by the ayahuasca administration, regardless of the dose, are represented in these reaction modules here. We had some metabolites related to the vitamin B6 metabolism, the pyridoxine and pyridoxamine. And this pathway is important for the biosynthesis of serotonin, dopamine, and GABA. We had also found out nicotinamide, which is related to the vitamin B3 metabolism, related to energy and inflammatory balance. And also another metabolites as isoleucine and 4-methyloxypentanoate related to BCAA metabolism, which is correlated to the energy production and redox balance. And all these pathways are important when thinking about depression. And we have seen that ayahuasca acts exactly on these important metabolic pathways. Here, this Venn diagram shows us the distribution of common and exclusive metabolites for each tested condition. So we got these 8 metabolites in common for the 3 concentration levels we tested, and another ones that were exclusively found for 1, 10, or 20 micromole per liter. Just highlighting the main results here, for the lowest ayahuasca concentration, we have seen metabolites related to the regulation of neurotransmitter precursors. For example, an increase in the concentration level of levodopa, which is interesting thinking about the treatment for depression. But for the highest concentration tested, we started to see some mechanisms related to oxidative stress and mitochondrial dysfunction. For example, by the decrease in concentration level of valine and palmitoylcarnitine. And this shows us the importance not only of characterizing ayahuasca, but also of determining its concentration in order to propose a treatment for a major depressive disorder. So moving to the last topic I would like to talk about, or more recent studies. I will show some results that were not already published, but I would like to show you a bit what we are doing right now in my lab. So we started to work with neurodegenerative disease models. We used these cells, the SHSY5Y cells, which are human-derived neuroblastoma. These cells are very interesting because they have some aspects that mimic human neurons, as we can see in this photo on the left side of the cell morphology, which has its neuronal phenotype. And these cells are usually described in the literature as models for studying neuronal function, diseases such as Alzheimer's and Parkinson's, and also to understand neurotoxicity mechanisms. And again, the same rationale we did for ayahuasca, we wanted to look for unconventional treatments for these diseases. And we found out that cannabis is being proposed as a symptom relief for Parkinson's and Alzheimer's. And then we wanted to understand better its mechanism of action in these cells. So cannabis produces also psychoactive compounds. The main ones are tetrahydrocannabinol, THC, and also cannabidiol, CBD. And these compounds are very interesting because they interact with our endocannabinoid system, which is responsible for the regulation of mood, memory, pain, and other brain functions. And the main thing here, again, as for ayahuasca, we are dealing with a natural product, a very complex one. So cannabis doesn't have only THC and CBD. It has another cannabinoids, terpenes, sterols, and also flavonoids. So the chemistry of cannabis is also very complex. For the cells we studied, we first tested some cannabis oils, which is what the patients take for their treatment. So for this study, we had 2 cannabis oils, an artisanal one prepared by a patient association here in Brazil, and another oil prepared by a pharmaceutical company in Colombia. Both oils were prepared in the same basis. The medium chain triglycerides is the matrix for both oils. We had a previous information of the concentrations of CBD and THC for both oils. As we can see, for the artisanal one, the concentration of THC is higher than for the commercial one, but for the commercial one, the concentration of CBD is much higher than for the artisanal one. And as for ayahuasca, we decided to normalize the concentration in terms of DMT. In this case of cannabis, we decided to normalize the concentration in terms of CBD. So we performed cell viability studies using MTT. We observed that the matrix MCT did not change cell viability. And for the cannabis oils, we observed these changes and we determined the IC50, the inhibitory concentrations for each case, for the artisanal and the commercial oil. Here on the left is a summary of these results. In blue, we have the percentage of viable cells. In red, the percentage of dead cells. For the untreated cells, treated with OC1 is the artisanal one, OC2 is the commercial one, and MCT, it is the matrix. So we can see that only for the artisanal oil, we have seen more relevant loss in cell viability. For the figure here on the right, we can see the results for fluorescence microscopy. The 2 first ones on the upside are for the artisanal oil, and on the bottom, the commercial oil. On the left, in both cases, the microscopy for the cells treated at 30% of the IC50, and on the right, the cells treated at 80% of the IC50. So in red are the dead cells, and in green, the viable ones. So we can see that the concentration of the oils also needs to be established and normalized before doing the metabolomics experiments. So for the metabolomics experiments, more specifically lipidomics, in the case of this work I'm going to show you, we decided to work at 25% of the IC50 to guarantee a lower toxicity and higher possibility of biological effects. We exposed the cells to the cannabis oils for 24 hours. Then we removed the cells, performed a 3-phase lipid extraction, and analyzed it by LCH-RMS. We compared then untreated cells and the cells treated with both oils, the artisanal and the commercial one. For the main results we found here, the main differential lipid class was the sphingolipids, and more specifically, the ceramides and the sphingomyelins. And why this result is interesting regarding Parkinson and Alzheimer's diseases. In the literature, it is described that for Alzheimer's and Parkinson's patients, they found an elevated ceramide level and a reduced sphingomyelin levels. The increase in the ceramides are related to an oxidative stress, neuroinflammation, and cell death. And the reducing of sphingomyelins are related to the disease progression and conversion into ceramides, which are more neurotoxic. And the cannabis oils, both cannabis oils, the artisanal and the commercial, both reduced the ceramide levels, the neurotoxic levels. So this was very interesting. And also for the commercial oil with a higher CBD concentration, we also observed an increasement in the sphingomyelin levels, the neuroprotective ones. And these results were in agreement with some we found in the literature. This study was published in 2021. And the authors here used an animal model exposed to CBD and a high-fat diet. And then they observed that the decreasing ceramides and the increasement in sphingomyelins. And this shows us that the cannabis oils act in very interesting pathways related to the neurodegenerative diseases. So besides the complex matrix of the cannabis oils, we also tested in the same cell line the isolated cannabinoids from analytical standards of CBD, THC, and a mixture of both. We also performed cell viability assays and analyzed it by LCH-RMS. So the first test for cell viability was regarding methanol, which is now the matrix for these analytical standards. We exposed the cells at 24 and 48 hours, and we observed that for concentrations up to 0.8, we did not observe loss in cell viability. And this was okay because we work with concentrations lower than that. For CBD, we observed interesting results because here we noticed enhancement in cell viability for concentrations up to 2.5 micromole per liter, a maintenance at concentrations of 5, and just a decrease in cell viability in concentrations higher than 10 micromole per liter. For THC alone, the behavior was again different because here the cell viability was maintained in concentrations of up to 10 micromole per liter. So here we did not observe an increase in cell viability, but a maintenance, which was very distinct. And for the mixture of CBD and THC, again, a different behavior. Here, in most of the cases, most of the concentrations tested, the proportions 1 to 2, 2 to 1, related to CBD, THC, we observed an increase in cell viability or a maintenance. So this was very interesting. And the cell viability results helped us to establish the concentrations we applied to the cells for further metabolomics studies. So here's summarizing the main results so far. This study is still in progress, but I would like to show you what we have found out so far. In this Venn diagram, we can see the distribution of common and exclusive metabolic pathways comparing CBD versus control, the mixture versus control, and THC versus control. So for the isolated CBD, we have observed alterations in the fatty acid oxidation pathway related to redox regulation and neuroinflammation. For the isolated CBD and the mixture with THC, we observed the involvement of the coenzyme Q10 biosynthesis, which is related to antioxidant pathways. For THC and the mixture, changes in the Krebs cycle related to the energy production. And for the isolated CBD and the isolated THC, changes in the vitamin B2 metabolism. Vitamin B2 is quite interesting because it is a cofactor involved in multiple metabolic pathways. For example, these ones I mentioned before. And these pathways are very interesting for further studies considering Alzheimer's and Parkinson's disease, these models. These studies, as I mentioned, are in progress. We have another ongoing project in our lab. We are also testing cannabis oils and the isolated cannabinoids in a differentiated cell line, the SHSY5Y cell. Here we differentiate these cells using retinoic acid to have a phenotype more close to the Alzheimer's disease. And we are also testing in U87MG cells, which has a glioblastoma phenotype. So we are also acquiring data from transcriptomics by RNA-seq of the same cell lines exposed to the cannabinoids, and our intention is to integrate these results using a multi-omics strategy. We are also evaluating the effects of the isolated CBD on the brain delivery of an animal model. These studies are almost finished. We are also comparing the effects at the cell level of DMT, this natural psychedelic, with a synthetic one, DMT. We are, regarding ayahuasca, we are also performing modeling studies to understand the interaction of the alkaloids of ayahuasca with human proteins using a molecular docking strategy. And we are also studying another psychoactive compound, which is psilocybin. And psilocybin is interesting, another natural source, which are the mushrooms. And psilocybin is a very unstable molecule. It is transformed in psilocin, which is the responsible for the psychoactive effects, and it acts on serotonergic receptors. It is being described in the literature for its good pharmacological safety, promising clinical efficacy, related especially to major depressive disorder, anxiety, and post-traumatic stress disorder. And it is considered as the psychedelic more close to medical applications. It has a very good standardization potential. So we are also performing our metabolomics studies on that. So I think my time is up. Just as take-home messages, metabolomics is a very powerful tool to elucidate molecular signatures of psychiatric disorders such as bipolar disorder, schizophrenia, and depression, and so on. And for the understanding of mechanisms of action of cannabis and other psychedelics, ayahuasca have shown a very good potential for the treatment of disorders such as depression, for acting in important metabolic pathways related to them, but its standardization remains a challenge for the community. And cannabis oil show this potential to modulate some metabolic pathways related to the treatment of Alzheimer's and Parkinson's, and further studies are necessary to investigate the effects of the isolated cannabinoids too, and also potential synergistic effects with other cannabis molecules, such as the terpenes and flavonoids. So again, I would like to thank Renata, for the invitation to be here, the EMN as a whole from the Metabolomic Society. It was a pleasure to be here. Thank all my lab members, current and former ones, our collaborators, the funding agencies, and thank you all for your attention. I'll be happy to take any questions. Thank you.
Thanks a lot, Alessandra, for this really, really nice talk. It was really interesting. We mentioned the question. I think do not hesitate to ask question in the Q&A section on the chat. The question will be read just after the presentation of our next speaker that I'm really pleased to introduce, Henrique Ribeiro. So he did a bachelor's degree in chemistry and a master's in analytical chemistry, and then a PhD in sciences at the University of Campinas in Brazil, then, as you already have heard about. And he also did a doctoral stay at the University of Turku in Finland about mass spectrometry, and particularly in metabolomics and lipidomics. And his research mainly focused on metabolomics, data analysis, bioinformatics, and he's using systems biology methodologies to, with the objective of aiding the diagnosis and understanding of diseases based on biochemical changes caused by different conditions in the human body. And currently, Henrique is a researcher, a postdoctoral researcher at Örebro University in Sweden, working in systems medicine, using, again, metabolomics and lipidomics to build outcome prediction models for traumatic brain injury. So thanks again, Henrique, to be here today. And yeah, the floor is yours.
Thank you very much for inviting me for this talk. Well, let me start sharing my screen. So I chose to put this title here, System Biology in Brain Disorders, because I'm going to split this presentation in two parts. One more turn it into psychiatric disorders, that is bipolar disorder, and the other one more to neurology, that is the works that I've been doing now with traumatic brain injury. So the first part is the metabolomic and proteomic data integration from bipolar disorder patients and their relationship with hemostasis. So just an overview of bipolar disorder. It is severe, and it's characterized by severe and persistent mood swings. And these mood swings, they alternate between maniac episodes or hypomaniac episodes depending on the type. Bipolar disorder type 1 is maniac episodes, and hypomaniac is type 2. And also with depressive episodes. The diagnosis is still performed through clinical observations using the DSM-5 form. And the symptoms, they start to begin on the beginning of adolescence, around 17, 18 years old. And as Alessandra said before, the biochemical and molecular mechanisms of the bipolar disorder, they are still unknown. So basically, on this work, the analytical methodology that we did was basically a GCMS-based strategy here, where we use 50 microliters of serum plus 150 microliters of methanol. We did the vortex, and then we put on the centrifuge. We did the derivatization with methoxamine and MSTFA. And then we added the internal standard, that is the pentadecanoic acid. The data set is composed by 8 healthy controls in 5 bipolar disorders, because these were the samples that we had with the proteomics data and the metabolomics data. And for the metabolite panel, we use all the 14 samples that we have for controls and bipolar to do the metabolite panel. So the data preprocessing was performed in MS-DIAL, and we used the FinLib and Go Metabolome database for data creation. And we filtered using the RSD filter over the 30% on the QC samples, and these metabolites that presented RSD over 30%, they were discarded due to the excessive deviation throughout the batch. So basically, the workflow that we did to integrate the proteomics and metabolomics data, basically we have the data from MaxQuant in proteomics and the data from metabolomics and MS-DIAL. In all the RStudio environment, we got these 2 data sets. We log transformed them, and in case of metabolomics, we normalized it using the internal standard. Then on metabolomics data set, we did the PCA. And after that, we performed the factor analysis to see which were the confounding factors of our data set. And after that, we proceeded to the multi-omics integration using the MixOmics package, where we got the canonical correlation analysis main contributors from the Diablo that is a part of the MixOmics package. And then parting from this integrated data set that has metabolites and proteins, we chose those with the contribution coefficient greater than 0.15. we performed a Spearman correlation and the first order partial correlation analysis, and then we did the integration network using all the metabolites, the proteins, and also some other demographic data. And from this metabolites that were selected, we performed also a beam plot to see the difference of the levels between them, between case and control. And with these metabolites, we performed a rich logistic regression, and we did a ROC curve to see which metabolites they could better explain to how can we differentiate between case and controls. So on the results that we obtained from this approach, so basically we did the principal component analysis, where we got a QC that was really clustered together, meaning that our analysis didn't have any kind of experimental shifts. And then after that, we did the multi-block SPLSDA that really revealed the main contributors, meaning the loadings that we use to maximize the covariance between X and Y. And these main contributors we exported for partial correlation analysis. These were on the factor analysis, we discovered the confounding factors, and we saw that the case control, it was what differentiated more, we explained more of the variance of the data sets, both on metabolomics and the proteomics data sets. And here's the partial correlation analysis. It's a little bit complicated to analyze all of these connections, but then we did one strategy here. We are looking especially into the case control node here. So if a metabolite and a protein, they're connected into the case control node, then we analyze this interaction between the metabolite and the protein. We obtained it using partial correlation analysis, and the proteins, when we analyzed what they were the effects in the human body, they were all related to hemostasis. And metabolites, they were also related to hemostasis, such as platelet aggregation, neuroinflammation, coagulation cascade, as well as gut-brain axis, the metabolites that we chose here. And it's interesting because one of the hypotheses of the biochemical alterations caused by bipolar disorder, it points out to a possible multi-system disorder. So this was really interesting to find out, like a connection with hemostasis and also the bipolar disorder. And these were the beam plots from the main contributors that were selected in the multi-block analysis, where we see that all metabolites here, they presented decreased levels on the bipolar group compared to the healthy controls, except for paracresol. And with these metabolites, we built a logistic regression model to try to find a molecular signature to differentiate bipolar disorder from healthy controls, where we reached this 9 metabolites panel, and the metabolite panel was adjusted for age, sex, and BMI. And interestingly, mostly of the metabolites, they were related to energy process like TCA cycle, beta oxidation, glycerophospholipids metabolism, and also glutathione metabolism. And one interesting case here is the paracresol, that it's a metabolite that's related to the gut-brain axis, and it's related also to possible alterations in the dopaminergic system, where it's shown in the literature that it can increase anxiety-like behaviors. So from this study, we concluded that there is a correlation between hemostasis and bipolar disorder, where we used partial correlation analysis. And this was corroborated by the individual analysis of the proteins, where on this article by our former member, Elisa, it showed that the proteins were also related to coagulation cascade and complement cascade. And the hemostasis process were also directly related to cardiovascular diseases. And the regional logistic regression, the model that we did, it showed a potential molecular signature to differentiate the bipolar disorder from healthy controls. We've selected metabolites mostly related to energy production. And the hypothesis that bipolar disorder is a multi-system disease, we think that it must be studied further to better understand how the systems are connected and associated to all the human body. And all this work that I just presented, it was published in 2022 in the Metabolomics journal. with the title Metabolomic and Proteomic Profiling in bipolar patients revealed potential molecular signatures related to hemostasis. So this was the first part of the presentation, where I wanted to present a little bit of the brain, but related to psychiatry. Now I'm going to the neurology parts, where is the work that I've been performing here in Örebro University. That is basically how can we use lipidomics as a tool to do outcome prediction for mild traumatic brain injury. So basically an overview about traumatic brain injury. Traumatic brain injury occurs when we have an external force that disrupts the normal brain function. And the most common causes are falls, vehicle accidents, sports injuries, and assaults. And it is one of the most common neurologic diseases present worldwide. And the patients here from TBI can be classified as mild, moderate, or severe based on the scale called Glasgow Coma Scale. And we need a more detailed characterization of the patients. This is required for us to have a better TBI management and also decision making. The most common outcomes evaluation method is by using the Glasgow Outcome Scale. We call it GOSE, the extended version. But even this outcome scale, it provides imperfect outcome prediction, and we don't have like it's not really good to explain all the cases. So basically, the Glasgow outcome scale, it comes from 1 to 8, where 1 is death, 2 vegetative state, and 8 is the best recovery, the full recovery. So basically, the overview of the study is something like this. We have the traumatic brain injury, we collect the blood 24 hours after injury, and this blood is sent to is from the center TBI cohort. The sample, then they are analyzed by a lipidomics analysis, and then we use machine learning to see if we can predict the outcome of these patients after 6 months. This is the main objective of the work here. The initial data structure that we have is composed by 3 batches that we had mild, moderate, and severe. And it was used where we used a modified Folk method to do the lipid extraction, and we analyze using a 1290 infinity system coupled to a 6545 Agilent QTOF. And if you want to know more about the processing steps, I tell you to refer to the article from Thomas, that is our former group member. And basically, the initial data structure had 357 lipids, where 239 are annotated and 118 were unknown. On batch 1, we had 948 samples and 20 QCs. On batch 2, 636 samples and 14 QCs. And batch 3, 559 samples and 11 QCs. However, if we do the principal component analysis where we get the PC1 and the injection order, we can see that we're a really big shift here between the end of batch 1 and beginning of batch 2. So we had these intra-batch issues, and we have like 83 samples and 2 QCs with shifts on the batch 1, and 129 samples and 3 QCs with shifts on batch 2. Unfortunately, we needed to exclude these samples because they had a huge shift and we could not use them on the analysis because of the huge shift that it has intra-batch. Then, excluding these samples, we use the QCMXP software to do the batch correction with these other samples. So after we removed these outliers, we had 867 samples, 18 QCs from batch 1, 507 samples and 11 QCs from batch 2, and 559 samples, 11 QCs. This was unaltered because batch 3 didn't have any kind of intra-batch problems. And after we did the batch correction, the filters, they were filtered based on the relative standard deviation above 30%, and the D ratio of 40%. And 278 variables, they remained after filtering. So this is before we did the batch correction, where we can see that on the PCA that the batches, they are separating here. This is not good for the analysis. And then after the batch correction, we see that the 3 batches, they converge together. And this means that the variance of the data set is mainly to the biological variation here, not to the batch variation. Then we subsetted the data to get only the mild TBI samples, because this is the type of patients that we are more interested in. So basically, we selected only the samples that had the GCS score above 13, and also the samples that had the available metadata, meaning the GOSI levels for 6 months after injury. And also we removed the individuals that they withdrew their consent, and also we removed the individuals that had below 18 years old that were removed from the final data set. So after the data wrangling, we got the batch 1 with 177 samples, batch 2 with 326, and batch 3 426. And how we are going to perform this data analysis? We're going to use batch 1 and 2 as the training set and batch 3 as the test set. So this is important for the next steps. So now we also performed lipid clustering using the cluster correlation with the covariates and proteins, because we wanted to see how these lipids, they were correlated with other covariates and also with some really established protein biomarkers like GFAP, UCHL1, and NFL, and some others. And from this, we saw that the lipid cluster 3 that mainly had lysophosphatidyl-choline, it presented the biggest correlation here. It presented a negative correlation with the proteins and a positive correlation here with the GOSE, with the outcome scale. And all these lipids, they were adjusted by the FDR correlated p-values. And this shows a really interesting thing, because we see on the literature that the NFL, GFAP, and all these, some of these proteins biomarkers, they presented on a higher concentration on the worst outcomes. And we also wanted to see, based on this, we saw we might see effect also on the choline on the patients as well. So we summed all the lipids that presented a choline. So this means lysophosphatidylcholines, phosphatidylcholines, and sphingomyelins that they present the choline heads. So we summed them all, then the intensity of these lipids, and then we performed violin plots, and we did the Juncker Terpstra test. That's basically a test that use it to predict a trend between several scales. So it's not like binary. So it's kind of a ordinal scale. And as the GOSI scale is an ordinal scale, this was really interesting to see how we saw that there is a true visible trend over the GOSI outcome scale. And then we also use this fact that the Glasgow outcome scale is an ordinal scale. Because of this, we chose to use ordinal logistic regression instead of regular logistic regression with dichotomization of each individual GOSI level. So basically, the workflow is like this. We did the NA droppings, we converted covariates and the outcome scale to the correct format, meaning we want to use the outcome scale as an ordinal scale here, and then we collapsed some GOSI levels that have fewer samples. For example, 1, 2, and 3, they have just a little bit of samples that were the mild cases, so we had to collapse them together. And also the patients that had GOSI 4 and 5, we also collapsed them together into the same group. Then we did the variable selection with ordinal elastic because we had a lot of variables, so we want to see which variables they were truly contributing to explain this trend, explain this continuum of the outcome scale after 6 months. So basically, we chose these lipids that were more associated with GOSI ordinally, and then we checked also if they behave consistently above these thresholds. And how we do that? We do the common odds ratio analysis that basically it checks if the individual lipids, they behave consistently across the GOSI thresholds. And finally, we did the proportional odds model where we chose some important covariates and the chosen lipids to be used, and then we validated these results on the test set and evaluated these results using ROC tests. So We selected 8 lipids on original elastic net. There was the phosphatidyl-choline with ether phosphatidyl-choline with 34 carbons and 3 saturations, lysophosphatidyl-choline with 16 carbons and 0 saturations, but also ether. The lysophosphatidyl-choline 20 carbons and 5 saturations, and lysophosphatidyl-choline with 18 carbons and 2 saturations, and also 4 unknowns that we probably will need to dig further to see which metabolites are these. So the common odds ratio analysis showed that they were behaving inside the error margin, that is the orange dashed lines. And all these 8 lipids were used to build the original logistic regression model afterwards. So then we did the original logistic regression of these lipids. We chose all these 8 lipids that were selected in original elastic net, and we used them together with the crash covariates, that is admission GCS, age, the pupillary reactiveness, and also the presence or absence of major extracranial injuries. And the Gaussian thresholds were something like this: the 1 and 2 against 4, 5, 6, 7, and 8. There is this one. 1, 2, and 3, 4, and 5 versus 6, 7, and 8. There is this one. 1, 2, 3, 4, 5, 6 versus 7 and 8. There is this one. It goes to 7. And then 1, 2, 3, 4, 5, 6, 7 versus 8. There is the last one. So basically, only on Gaussian 6 we see that the results of the adding up the lipids to the model, it was worse than just the covariates. But especially on Gaussian 7 and 8, they are the most complicated ones because they are really difficult to differentiate between themselves. We saw like a really good increase from 0.673 to 0.714 on the Gaussian 7, and on the Gaussian 8 we saw an increase of 0.061 to 0.683. It's still not ideal, but we can see that adding lipids up to the prediction model, it can help us to further understand and explain better these prediction models. And also, we got all these 4 from these 8 lipids. 4 of them were annotated. So basically, we selected these annotated lipids, and we did the same thing that we did on the sum of the colon. We did the Juncker trend test for the selected annotated lipids. And we saw that all of them presented a p-value less than 0.01 on the trend test. And they are all, interestingly, all these 4 lipids, they were already reported in the literature, and also with the same trend. The higher concentration on the better outcomes. And lysophosphatidyl-cholines and ether-linked phosphatidyl-cholines, they are linked to neuroinflammation process in the brain after the injury, and they can act as mediators and signaling molecules which can help us to understand better the physiopathology of the pathophysiology of the TBI, and help us explain how we can see how a patient can behave after 6 months. This is the main working power behind this work. So basically, on the conclusion from this work, this is going to be published really soon, I hope. So the batch correction method that we use, we're using the QCMXP, improved the overall data and corrected possible drifts between the 3 batches. And we showed on the lipid clustering that cluster 3, which contained mainly LPCs, presented a significant negative correlation with protein biomarkers and a positive correlation with the Gaussian outcome scale. And on the JT test, we showed this visible trend on the colon levels over the Gaussian outcome scale. And using the original logistic regression, we showed an increase on the AUC when compared to the models using only the crash covariates in some of the dichotomization. And from the 8 lipids that we selected, the 4 of them that were already annotated, they presented higher intensities on better outcomes and a p-value less than 0.01 on the JT trend test. And the LPCs, they are related to neuroinflammation process in the brain, which can help us explain this relationship of this trend over the outcomes from the TBI patients. Well, finally, I need to thank a lot of people. And first of all, thank you all for the Early Career Members Network to invite me for this presentation. I want to thank you also, all my friends from LaBIOmics, and especially Professor Alessandra Sussulini and Elisa that helped with my PhD work. And also the guys from Turku Bioscience, Professor Matteo De Sicti, that he also is, he's a professor in Turku Bioscience and Örebro University. So he was the one that hosted me there when I did my research stay during my PhD. And also I want to thank Alex Dickens, Parto, Marina Alves, and Matilda Krasholm, because of they all of them worked together with this, not only with the old data set, but also with the new one. And also I want to thank all of my co-workers here from the System Medicine group, Professor Matteo Recic, Professor Tulia, and all my other friends from System Medicine group. And also I want to thank you all people from Center TBI, especially Professor Andras Bucchi, Professor David Manon, Andrew Mas, and Veronique de Kaiser that helped us with this huge project that has a lot of samples, a lot of things we need to analyze here. And also I want to thank to all my funding agencies and to VR, the Swedish Research Council, for being the main important funding agency here from this TBI work from now. So with this, I'm finished. So if you have any questions, just drop on Q&A and I'll be glad to answer them all. Yeah, that's it.
Thank you very much for your great presentation, both of you. I think now we can start with some questions. Let me check the first one. So, what are the main methodological challenges in studying the effects of this plant substance in patients with bipolar disorder and depression?
So, thank you for the question. It's not really a problem with the methodology here in our lab, the metabolomics part, but with the clinical setting, because it is not easy to have a proper clinical setting to receive these patients and to offer them an alternative treatment. We need to get a close interaction with the people from the medical school, and they need to have a proper clinical setting to receive these patients. So it's not so easy. Here at the University of Campinas, we don't have this proper clinical setting to receive the patients, but we have a collaboration with the University of São Paulo in Ribeirão Preto. There they are starting to study the effects of ayahuasca, first in a control population with no psychiatric disease, because the side effects here will be expected to be lower, right? Because some people don't respond so well to these treatments with psychedelics, and they need to be accompanied by doctors. So we do need these clinical settings, and this is the main challenge in this type of research when thinking about applying to the patients, and that's why we have chosen to work with the cells, so we can understand the metabolism at an in vitro model.
Yeah, I think this answered the next question, which is like, what are the biggest challenges in translating these metabolomic findings into clinical practice? So
yes, it answers, because it is the main problem is that it's not the part of metabolomics. For metabolomics, we are okay, but the clinical setting and all the ethical aspects involved too, because it's not easy to get approved. Just recently in Brazil, cannabis, for example, was approved for more broad medical purposes. So we have also, besides the clinical setting, also the legal aspects that make the research a bit slower when thinking about patients.
The ayahuasca is a treatment for depression, or it's in the process of becoming a treatment?
Is being studied as a treatment, but this first studies was just performed with the single dose of ayahuasca. So people had depression, they have taken many treatments and do not respond. And just with one single dose, they had an effect of about almost 1 month. But this study stopped there. So more studies are necessary because they needed to understand the doses should be repeated or not from each time. So these are main questions. But the interesting thing that in ayahuasca rituals, usually people that take ayahuasca regularly, they do it every 14 days. It's not every day, it's just every 14 days. So this is also a previous knowledge. So this might be translated to the clinical, but this needs more following up studies. So
you want to go for the next one, Thomas?
Sorry?
Yeah, there is one here that asked how were your samples processed for metabolomic study, but which one? Can you specify? Because there were several different, mine and from Alessandra. So each one of them use a different data processing strategies.
Talking about this, one for you, Henrique. What are the main challenges in this integration between lipidomics and proteomics? Because I already tried once, but because of I think the intensity of the peaks in proteomics and lipidomics are quite different. I tried this integration, but at some point they were separating too well. And I said, I'm not sure they are integrated really well. So I think, what are the processes you use to make this better?
Oh, on the proteomics side, I remember because the proteomics side was Elisa that made the other former group member. So as far as I remember, she did the regular normalization with the proteomics data, and then I used the normalized data. Just normalize it. I haven't done any kind of transformation, like log transformation and anything. I did that myself before in order to be sure that everything was following the same protocol. So I did the log transformation with both of them, but on the metabolomics I had to normalize. So after all the data was log transformed, then I put them on the mixomics package. And then on the mixomics, we selected the proteins and the metabolites that they had the contribution score higher than 0.15 plus or positive and negative, because we also want to see the negative correlations as well. So if we just select the positive ones, we exclude the negative correlations. So since we want to see both the positive and negative, we selected both cases. And honestly, I think that's all I remember, because that was I haven't done much transformations on the data, because the data that I got was already pre-normalized before. So basically, I think was that.
Right, I have one more. I think for Alessandra, the comparison between bipolar disorders and schizophrenia showed most sphingolipids. Do you have like an idea what is lipids? Is like ceramides, sphingomyelins, or you don't have yet? Because you said you're annotating there in the next project, but I'm not sure if you already have some information.
Yeah, in that paper I have shown, we have some. I just summarized grouping together with the class, but we also found some sphingomyelins mostly.
Okay. And the ceramides that you found in your cannabis oil, they are like more long chain, mid chain, or very long chain saturation. Do you have this information? Because some disease.
No, I don't remember by heart. I need to look to our data. I just summarized it there, but
Yeah,
I don't have this information.
They show more the long chain are the bad ones, but now the very long chains are also appearing. So I was just curious to see.
Yeah, but I don't have this information right now. Sorry.
What motivates your lab to try to find these alternative treatments to plants instead of drugs, which is the normal treatment?
We wanted to see another alternatives. The first interest was in ayahuasca because it is like a cultural product, not just a natural product, just not any natural product. Because we are here in Brazil, we have this culture. I wanted to explore this better, this traditional knowledge. That's what motivated us to started to looking at it. Because the reports of, for example, the people probably don't need to take it every day. For example, if you take a treatment every 14 days, it would be great, because it is not good to take medicine every single day. Sometimes you forget, so it's not so good. And there are many aspects here. For example, the low collateral side effects, because sometimes they have some side effects, not so good, the conventional medicines. And this everything motivated us in this sense, the cultural part, and also the probability of not need to take a medicine every single day, have lower side effects. So we wanted to explore that. And for cannabis, it also is also a natural product. It also has lower side effects as many drugs for Alzheimer's, for example. I was motivated to study Alzheimer's back at that time because I had some cases in my family. I was very interested in studying Alzheimer's, novel or alternative treatments. And this was very interesting to use your knowledge in metabolomics to further explore these other possibilities besides the pharmacological, more known ones.
I have one last one here for Henrique. The idea from like you're searching the lipids that are related with the trauma, like the brain injuries. The idea is maybe related to the scale, because then you have something to measure, not just to use like a subjective scale. Is this kind of the idea?
Exactly, because we want to do something less subjective, exactly. Like the same way with the psychiatric disorders. We want to have something that you can just do a blood test and see, oh, you have like a high risk of after your trauma that you are not going to behave well. So basically, the main focus is that to see if we can also allocate properly the right resources to the right patients. This is most a decision-making tool, to be honest.
Yeah, and I imagine, for example, if a person had a brain a trauma, and then maybe if some of these lipids are like overexpressed, then there can be a relationship with other types of diseases and like brain disease.
Yeah.
Yeah. I think it's all for me. Do you have anything else, Thomas?
No, I think we answered all questions from the chat. I would have a more technical question for Henrique, if it's possible for the last minute. Why are you using ordinal models rather than more classical ones like generalized linear models, for instance?
Well, this was a suggestion for Professor Ewald Steyberg. He was one of the professors that worked together with us on Center TBI cohort. And this was actually, if you think about it, it's a ways to use other regular models like a logistic regression model, usual one, and then dichotomize, because then we lose all this originality of the scale, right? If we use a ordinal scale on an ordinal model on an ordinal scale, then we are truly seeing the continuum of the scale instead of each cut point. So I think it makes more sense for this scale to use the ordinal scale.
Yeah, no, I agree. It's not my domain, but
there are some works from Professor Ewald that are really good and really informative on the ordinal scales.
No, thanks a lot.
Professor Steyberg.
You want to conclude, Renata?
Sure, I'll just share here my screen to show everyone the. Let me here. So, thanks everyone for coming to this webinar. It was a great pleasure to receive these great talks from Professor Alessandra and Henrique. Just to show you here, if you want to connect with us, we have our social media, the EMN. So we are most active on LinkedIn, but we also have the Blue Sky and other ones. If you want to send us an email, feel free, and this email here. And next month, we have one more great talk. It will be Professor Diego Padillo Sanchez. He was going to talk about archaeometabolomics, a new way to delve into the past. It will be on the 26th of March. It should be a very great talk. It's a very different topic. And one last thing, on the 31st of March, we will have a Korean Metabolomics Networking event. So they will basically talk about developing a scientific identity as an ECR in metabolomics and beyond. So if you are an ECR and you are interested, just put in your calendar this date. We will start showing in our social media all these events. So check there. If you're not following us, please do so you can have all the information and also you can receive the newsletter. Okay? So again, thanks the speakers for accepting the invitation. Thank everyone for coming and see you next month.
Thank you.
Thank you. Bye-bye.
Bye.
Thanks a lot.
All right.